Sorry, you're quiet. Let me turn you up.
Don't forget to hit record.
I did. Thank you
Okay.
for the reminder, though. But I did actually hit record. So because I know people, where everyone go? Because some people will want to look at this for. Make me big. All right. Stop sharing. You don't need to see my screen. So nothing. No one has any questions about anything. You're completely ready.
Hi.
Hi.
I'm just confused about the differences between bound ribosomes and like free ribosomes.
Okay, what do you think the difference is?
Well, I mean, like, I'm just confused where their proteins go. That's what I mean. Because like I know that bound ribosomes are attached to like rough ER, and like free ribosomes are like, are just like in the membrane inside.
Not in the membrane. They're just in the cytoplasm.
Yeah.
They're just free inside the cytoplasm. Most eukaryotic cells do not have a lot of free ribosomes.
Okay.
Mostly bacteria have free ribosomes, prokaryotes. So in eukaryotes, most of the ribosomes are bound. There's no difference to what their function is. So their function is to make protein. If a ribosome is bound to endoplasmic reticulum because it's in a eukaryotic cell, which remember, plant cells, animal cells, fungus, protists, algae, all those are eukaryotes. As soon as the messenger RNA begins to be translated into protein, that protein, those amino acids just get poked and sort of stuffed into the end of the hole, the lumen of the endoplasmic reticulum. Remember, the ER is like a tube. So those ribosomes that are bound, as soon as the amino acids start to be synthesized, they're going to end up going right into that tube of the ER until the protein is finished. Then the ribosome lets go of it, it dissociates. This doesn't float around in big pieces, stays close to the membrane, but it dissociates. And that protein then is inside the endoplasmic reticulum, and it's going to go along and be folded. That's what we call protein processing, right? It's going to get its secondary, tertiary, quaternary structure. And in some cases, it's going to be, you can think of it like being polished. Some parts might get cut off, rearranged, folded. And then in the Golgi is where that protein is actually going to get its other organic molecules attached. The sugars, lipids will get attached in the Golgi, and the sorting happens in the Golgi. But the actual folding of the protein happens in the endoplasmic reticulum.
Thank you.
You're welcome. Hopefully that's better, not worse. Anybody else? Yes, no, maybe. Okay, so just a little bit about the test then. It's, I believe, a little bit shorter. How long was the last one? I'm getting my classes confused. I think it's a little shorter, not sure. And expect complex questions. Okay, so just like before, there'll be, now that we're into more of the actual, what I would call the meat of the semester, I'm going to ask you more complicated things rather than just, can you name this? Can you identify this? I'm going to ask you things, how is this different or similar than that? So for example, even on identify question. So trying to help you guys with test taking, because you're not always going to be online, right? So there's this set of categories, it's called Bloom's taxonomy, and it's how exams are arranged and how questions are assessed on how difficult they are. So the very basic level of Bloom's taxonomy, level 1 is knowledge, okay? Knowledge / comprehension. And that just means, do you know this definition, or do you know what this item is? And then it gets more complicated, right? So do you know it? Do you understand the fine details of it? Can you use it in a new way? That's the analytical / critical thinking levels. So For example, on this exam, there will be questions that will just be, what is the function of the endoplasmic reticulum? And there's 4 choices, right? That's an 8th grade question, high school question. These questions will be more like, which of the following is not a function of the endoplasmic reticulum? Or which of the following organelles are mismatched with their structure, with how they're set up? Or which of the following organelles are mismatched structure to function? So there's going to be a lot more, it's going to be a lot more complicated than just define this, or do you know what pH is? So I would study, I would check out those dynamic study modules that are affiliated with mastering. So they're not things that I assigned, they're just things for you to use to study. And I believe they're in the study tools area, and there's flashcards if you haven't made them and you feel you need them. And then there are also these dynamic study modules, which kind of help you focus where you're weak, right? So they help you spend more time on the concepts that you know less about. But bear in mind, it's only 2 chapters. So all of the concepts that we've covered in 6 and 7 are going to be treated pretty much with equal weight. Like there won't be more of the test about chapter 6 than chapter 7. It's pretty balanced. Still no short answer. Not sure. There might be matching. Don't know. And there will be several of which the following is not, or which of the following is the best choice of. So it's a little, be careful when you read the question, really read the question. The questions will be the same, they'll show one at a time. And I would just remind you, there's a lot of academic dishonesty going on. I understand it's online course. There's this undeniable urge to be like, well, I don't need to study that much because I can just look it up. I would remind you there are methods that there are things that I can do so that I can see what you've done while you're on the Canvas page. It records every time you leave the page, and I can see that. It also records, obviously, how long it takes you for each question, how long it takes you to answer each question. The timing will be the same, be 45 seconds per question. And I would just remind you that you need to know this information for your next class. So whatever the reason is that you're taking this, unless you're taking it as a lab science elective and you're an education major, for example, you actually need to know this stuff. So if you don't know it, it's not really hurting me. It's going to hurt you later because you're going to have to learn it at some point. So I would just remind you, if you are being academically dishonest, it is cause for removal from the course, and that's in that first day handout. I will give you an F on the exam, and I can ask that registrar to actually just take you out of the class. If it's discovered that you're doing this in multiple courses, you'll actually be removed from the college. So you need to think about if that's worth it to you, just to get maybe a 78 instead of a 72 on an exam. It's a long semester, guys. We're at the end of the first month. We have 3 more months to go, basically, at least, right? All of October, all of November, and all the way to December 21st, technically. So there's lots of time and lots of opportunities for you to improve your grade. Don't panic. The exams are 70%, but the mastering homework and the labs together combined are 30% of your grade. Do a good job on those. Turn them in when they're due. That increases your grade, right? It's a weighted average. So 70% is your exams and 30% is lab and mastering. So the labs, I'm probably going to start them. I'm hoping to start them this Friday. And like I said, I'm going to make little videos about what to do. You're going to need to do the lab exercise and then take a photograph of yourself with your results. And then I'm going to design lab quizzes, probably not for each lab, but maybe 1 or 2 labs together. And those lab quizzes then will be based on what you were supposed to learn during the lab activity. I understand, I'm aware that in the box comes the instructor's guide, which is important for you guys to read because it'll give you all the background information. And then all the answers are in there, of course. So I would encourage you to try to do it without just looking at the answers, but the answers are all there, so you should be able to get them right. And then, like I said, I'm going to design a different lab quiz that asks you about your procedure, what results you got, and then, of course, some variation of those questions that you had to answer. So I have to make the lab worth doing, right? So it has to count for something, but it's not something that you should have a lot of anxiety over, right? They're mostly fairly simple activities. I didn't pick them, but the folks that did pick them tried to pick things that are simple enough that you can just do them at home, obviously. I know for the microbiology kits, there are things that have to be refrigerated. Check your kits, make sure. I don't know that there's anything in our kits that has to be refrigerated, but if there is, please go through and find it and just stick it in your home refrigerator. It'll be fine. It's not toxic, it's not dangerous. And I can tell you for the DNA extraction, there's ethanol, I think, in that one. And I'm going to ask you to put that into your freezer because it precipitates the DNA a lot better if it's super cold. The colder, the better. So when we get closer to doing that actual lab, I'll remind you, but none of the things that are in these kits are hazardous. And if they're even mildly, even I think distilled water comes in a Ziploc bag, right? It's in the bag, in a bottle, and the bag has Ziploc, so you can just put them in your refrigerator without worrying. My kids have been, me too, but I would check it. I think what'll happen, Colleen, is if it's something that needed to be refrigerated, it might not work as well as if it had been cold, but unless your house is like 80 degrees, it should be fine, right? So most of the experiments are for room temperature. So I'll just say that. I know in the microbiology kits, that's what made me think of it. I know in the microbiology kits, there's things that you have to put in the refrigerator or they'll go bad in a week or two. But I think it said their shelf life was like 2 weeks without refrigeration. So you're still in the clear. All right, other questions people have about the pending doom? Yes, no. I'm trying to remember. We completed chapter 8. Oh, which is the trickiest subject matter for the exam. For this exam, like I said, I would say the trickiest part is going to be reading the questions and being sure that you know what the question's asking before you just choose an answer, right? So that's probably, I don't know. I think for most students, the hardest part is the sheer volume of information because there's so many fine details in chapter 6. And then, of course, the functioning of the membrane. Some people have trouble with the solutions that we had talked about before. Hypertonic, hypotonic, isotonic. Do you have a question? You keep showing up as my big screen here. Do you have a question? You can either type it in the chat or just go ahead and ask me.
Oh no,
Oh, wait, I muted myself. That wasn't good. All right, so anybody else? Anything else that you're worried about, concerned about? I'm excited. I think I might actually get to see a kit. So later this week. I shouldn't say that before I know, a lab kit. And then I'm pretty sure there are 7 labs in the kits. And so my intention is to set it up so that every Friday from this Friday or next Friday forward, there'll be a little presentation on how to do the lab. And then those of you that have the kit can do it. Those of you that don't have the kit, when you get them, you can watch that video and then do the experiment. I don't think any of these experiments take longer than 20 minutes, half an hour, maybe 40 minutes at the most. So I think it's, like I said, it's not something to have a lot of anxiety over. We're just going to do them as best we can. Whether your results turn out well or poorly, you're going to hold them up against, you know, like a white piece of paper if they change colors, so I can see the different colors, and then send that to me and answer the questions. That's going to be about it. All right, so that's tomorrow, and then I think the rest of chapters 8, 9, and 10 are built out in the next module for you. I do need to check the dates on the library assignment because I think the going to the webinar is over. I'm not positive. I want to check that, make sure everyone's gone to the webinar. I'll post my video, my short little mini video on how to do the library assignment, and then we'll start talking about that probably next week. I did put that on my calendar. I like to wait until you guys know a little bit about biology and a little bit about some of the vocabulary before I just say, hey, go read some research papers, because it's really hard to read them if you don't know anything about anything. If we at least get through the cell, the membrane, and metabolism, a lot of the vocabulary then starts to become a little familiar. But that's good, that's all I know. Anything else? Any other questions? Yes, could we go over the functions of peroxisomes, please? Yeah, sure. So not all cells have peroxisomes. It's a little deceiving that your book lists them as a completely separate organelle. They're super specialized compartments, and the reason they're called peroxisomes is that they contain hydrogen peroxide, which is toxic in and of itself, but it's a huge oxidizer. So when you have a molecule that, like particularly something that has a lipid in it, and you're trying to break it down, a lot of times you have to oxidize the lipid. So you have to oxidize the fat. Again, in Chem 2, you'll learn about beta oxidation in particular. So if you think about any old or tired organelles that the cell needs to get rid of and then replace, the cell's going to recycle as much as possible. So it's going to put that organelle, I think in your textbook, it's an old mitochondria. It's going to put it into the peroxisome, and the peroxisome is going to break down the phospholipids into smaller bits and break down that inner membrane of the mitochondria, and maybe even break down the DNA that's inside the mitochondria, and then release that into the cell, into the cytoplasm, usually in a vesicle, and that's going to be taken back to wherever the mitochondria is assembled to make a new mitochondria. It's going to be recycled if it's possible. So like I said, in your book, they show a peroxisome that has a mitochondria inside of it, and then they mention some other specialized functions of peroxisomes. You can skip that, right? Because anytime in this textbook in particular, because it is, if I may say so, frickin' encyclopedic, anytime there's only like this much, or a column or part of a column on a page in a big picture, that means we don't really know, right, still. So it's what, 2020, and that's all we've got about peroxisomes, right? So it's a good chance for me to point out to you that as encyclopedic as the book seems, there's still so much that we don't know, right? That science doesn't know about all the parts of the cell and how they work together. So on that same page, like on this page next to that, this starts the parts about the cytoskeleton, and absolutely. And I look at those pictures of the cytoskeleton, and in microbiology, for example, in prokaryotic cells, we always say prokaryotes don't have a cytoskeleton. And now the newest edition of our textbook says they do. There's actin filaments in there, which is, you know, analogous to, what did you guys have, the microfilaments. So the more people know, it just keeps getting like more complex. All right, so someone asked about pinocytosis and receptor-mediated endocytosis. Sure. So you'll see those at the very end of the chapter, I believe, of chapter 7. I'm just flipping ahead here to look at the pictures in my book, which the pictures, those 3D pictures, I think are super cool. So there's an animation for this in your book as well. And if you look at the dynamic study model modules, I think there's a whole section on this. Yeah, I have my board set up, so I'm just gonna pop over there and I'm gonna use a couple different colors here. To make this as big as possible. Pretty good. Okay, so all of these, the things that you asked about, they are, remember, they are all active transport. And I know that's a little bit confusing because they're not showing you any ATP in that process when they show it in your book, right? But remember, just as a list for active transport, we would have ion pumps, like the sodium potassium ATPase, right? Or a hydrogen pump, like ATP synthase. And then we would have these sort of specialized bulk, what we would call bulk transport. And the first two, the first one is not very specialized, right? So pinocytosis, there's 3 types of bulk transport, meaning we're bringing in a whole bunch. It's like going to Sam's, right? Or Costco or BJ's. We're bringing in a whole bunch. Pinocytosis, phagocytosis, and receptor-mediated endocytosis. I'm going to put C there. It says yes. That's a long one. Endocytosis. So pinocytosis is, we call them pinocytic vesicles because they're very tiny and they bring in fluid. So it's usually, people say it's analogous to like the cell drinking, right? And it's going to bring in mostly extracellular fluid plus a few small things, few small molecules. So you'll see the picture in your textbook. There's like this. Pinched off, and then maybe one that's less, that's just like starting to form, right? So if this is a vessel, right, where like a capillary, you can think of it like a capillary where fluid is flowing by, or if it's, you want to think of it like as the extracellular matrix, between cells there's fluid, and then the cell itself, like the phospholipid bilayer membranes of the cells would be here, right? So I'm just going to draw these. These are supposed to be the cells. So I don't have much of a tip on my marker here, but think of this as the phospholipid bilayer of cell 1, cell 2, and cell 3. And these are, I don't know, they're skin cells, they're liver cells, they're whatever. They're cells in the small intestine. And everything that's floating by here, and I'm just going to make them be dots. Right? So I've got some blue dots and some, it won't show very well. So let's see, I have an orange. So I put some blue dots on there and I'll put some orange squares on there, right? And so this is just fluid that's literally moving and flowing past this cell. And the cell's just like, oh, I'm gonna make a little vesicle and see what I get, right? And so various amounts of whatever's passing by is just literally randomly being absorbed into this cell, okay? Peptide hormones, whatever it is, whatever it is. So the important part is that it's coming by in fluid, and then the cell is pinching off into little vesicles, and then this vesicle, so the nucleus, of course, would be much bigger here, right? So there's the nucleus, the nucleolus, right? And like the mitochondria over here. A very good drawing, sorry. Some endoplasmic reticulum, right? And the cell's going to pinch this off, and then it's going to take that vesicle once it's pinched off to wherever it needs to go in the cell. Phagocytosis is particles, we say, because we're talking about bigger things. And the example is almost always with phagocytosis. The example is almost always a protist engulfing a bacteria, because it's bringing it in as food. And that's the picture that's in your book. It's an amoeba, is the protist, and it's literally engulfing this whole bacteria that is maybe 1/8 the size of the amoeba. And the bacteria, the amoeba is going to ingest it into this phagocytic vesicle that then is going to fuse with a lysosome. And the lysosome, the enzymes in the lysosome are going to break down what's inside that phagocytic vesicle. And then the cell's going to use what it can use. So if there's glucose in there, there's starch in there, the cell's going to use that to make ATP. And the rest of it's going to spit out. That would be exocytosis. And then the receptor-mediated endocytosis is the most sophisticated of all, in that we have our cell. There's the endoplasmic reticulum there. I did a better job this time. Golgi over here, mitochondria over here, right? So in the case of receptor-mediated endocytosis, the cell isn't just randomly taking whatever comes by. The cell specifically on its surface has proteins, and I'm going to make them have a sort of very squarish, and they're located all over the cell, so the cell can get this substance from whichever direction it comes in the extracellular fluid, because remember, extracellular fluid is going to be bathing this whole cell in those little junctions and spaces between the cells. Receptor-mediated endocytosis, you need a receptor, which I made in orange, and then you need a ligand that's going to bind to that receptor. And my ligand, of course, is going to be a square now, because I've made my receptors have squares shape for their. So this is the receptor. And then the cool thing that happens is once the ligand binds to the receptor, and it happens to more than one of them, the receptors start to cluster together. So time lapse. This. So it's still the same cell, nucleus, endoplasmic reticulum, mitochondria, whatever. But the cell starts to pinch off groups of receptors that have bound ligand. That's why it's called receptor-mediated endocytosis, because only receptors that have bound the purple square are going to be put into this particular vesicle. I'll draw those in there. Okay. And then actually this vesicle gets a special protein coating. I'm going to use yellow. I should have used the brown, but I'm going to use yellow. That tells the cell, hey, when this becomes a vesicle, this is not just a random phagocytic vesicle. Don't send over a lysosome, because this has like liquid gold in it. This has stuff that's super important to the cell inside of it. And almost always, so I'm just going to draw that protein again. And I'm only going to put one receptor in there because that's all I can fit in my drawing. But I'm going to put my square in there. What happens in here is the receptors and the ligands are separated from each other. So there's my receptors, but now they're not bound to the ligand anymore. And then the ligand gets put into a smaller vesicle that gets transported usually to the nucleus, because usually these are molecules that need to tell the nucleus to do something. So this is exaggerating the size of this vesicle, obviously, because it would not be as big as the nucleus. But it's going to the nucleus, and it usually has proteins in it that are going to tell the cell, hey, make this DNA, make this RNA, make this protein. Time to divide. We're getting too big. Super specific. If this receptor is confronted with, oh, here I can use my line, triangle ligand or circle ligand, they don't fit in the receptor, so they don't bind and they don't get taken into the cell. Only the things that fit perfectly into that receptor get taken in. I usually tell people, think like right, if you play football, American football, right? So you're trained and you're a receiver. And I like to use this example because receiver is, receiver and receptor are the same. So I would say you're a receiver and you're programmed to catch footballs, right? Could you catch a soccer ball, basketball, hockey puck, a wiffle ball, baseball? Of course, right? But you're looking for footballs. And if you're in a game and you're playing, you're expecting a football to come to you. And you're only going to catch the football. It's like that, right? So the receiver, the receptor, is only going to catch footballs. It's only going to catch, in my example, purple squares. And it's not going to catch brown triangles or yellow circles or whatever else that I would have drawn, right? It can only fit one thing. And then it's going to take that message and take it to the cells, usually to the nucleus, and tell the nucleus to do something. We are not going to spend a lot of time with chapter 11, which is all about how those receptor molecules that get bound actually trigger events in the nucleus. Go to cell biology, learn all about it somewhere else, in my opinion. We have enough to do in BIO1. You're welcome. Probably longer story than you wanted, but. And tell me, how do I say your name? Is it Nahiba? Nabia?
Nabiha.
Say it again for me.
Nabiha.
Nabiha. Okay. Thanks. All right. Any other questions for the pending doom? Doom on you. No? Yes? All right, then. I'm out of here. I'll see you guys on Wednesday. I believe we finished chapter 8 already, and there are videos up for chapter 9, I think in parts. So there's a little bit that I will probably ask you questions about from chapter 8 to see. I know it's coming soon, but try to stay a little bit every day, right? All right, good luck tomorrow.
Is there going to be some sort of like exam for chapter 8? Because I know chapter 9 has like a Pearson test.
Oh, no, I just put that on there for chapter 9 because it's hard. And I wanted to give you like a pre-test test for that one.
Okay.
So no, that only shows up for chapter 9, actually.
Okay, so then the next exam will have chapter 8 in it.
It will have, the next exam will have 8, 9, and 10
Okay.
all together.
Okay, thank you.
But I'm trying to get you to get in really deep into chapter 9, because if you can wrap your head around most of the concepts in chapter 9, and then you read chapter 8 again, you're like, oh, that's why we talked about that.
Mm-hmm.
And it helps you when you get to chapter 10, you're just like, oh, I kind of already know this. So chapter 9 is key. I don't know how else to,
Thank you.
You're welcome. All right, I'm gonna stop.
Hi,
that.
just one question really quick.
Sure.
Okay, our quiz tomorrow is going to be on all the things on the outlines for chapter 6 and 7, right?
6 and 7, yep. And in those videos for 6 and 7, yep. And in the book for 6 and 7.
Okay,
Right?
thanks.
Focus on what's on the outline, obviously, but I can never say, because the textbook is the source for all our material. I would say, I do have written down in my notes, and I should have said this on the, before I stopped recording, the co-transport, I have written skip, and I probably said that in the video for chapter 7.
Okay. Thanks.
You're welcome. Good luck.
Thanks.
All right. Let's go back to gallery view so I can see who else is still here. Morning.
Could I talk with you for a little bit after everybody leaves?
Absolutely.
Sweet.
I am going to try to make this go back to, here we go, to where I can see everyone. And can I make that large? Oh, what does that button do? Let's do this, gallery view. There we go. Now I can see everyone. All right, guys, if you're not hanging around for a question, I'm gonna put you in the waiting room. So I'm gonna talk to Angelina for a minute. I'm just giving her a second. Somebody else. All right, Mike. All right, he's gone. I'm gonna pop her back into the waiting room. Okay, there. Everyone else is in the waiting room.
Okay.
What's up?
I just wanted to let you know that this past Thursday, I actually broke my arm. And I'm gonna be having surgery this Thursday on it. Yeah, I broke it pretty bad.
Can I ask what you were doing, or is that none of my business?
Roller skating accident. Yeah, me and my boyfriend, we were out getting some fresh air at the park, and I'm not used to outdoor skating yet, and I went a little fast, and I couldn't stop, and I fell right on top of it. So I have a nice compound fracture on my radius, and a bit of my ulna is
Oh my
broken.
gosh.
So yeah, it's yeah. But I just wanted to let you know that I am right hand dominant, so I might be a little slow doing some of my work. My mom's going to be helping me with it.
That's fine.
Of course, I'm going to try and get everything in before the due date. But like I said, I'm having surgery on Thursday. It's outpatient. Doc said I should be good, right after surgery. But I just wanted to make you aware that I might be slow getting some of the assignments in.
That's fine, Angelina. That's why there's so many assignments, is because for some people, and they're marked 5% off a day that you're late. So
Yeah.
hardly anything. So if there's ever one that you miss, don't freak out. Just do it anyway. And then don't tell anyone. I have a feeling you're probably going to be one of my A students. So I probably won't need to adjust your grade at the end or anything. It'll probably just be in the 90s. But nonetheless, right? So
Yeah.
try to get your work done. You'll be fine.
All right. Thank you for that.
You're welcome. Good luck with your surgery.
Thank you. Have a nice day.
You too.